Alright guys, let's get started with the topic for today. So today's lesson, we're going to look at specific enzymes, three for DNA polymerase. What you guys don't know yet is that there's two types of polymerase, right? You have learned about RNA polymerase, which is the polymerase that generates mRNA for transcription, translation, and so on, right? So far, you haven't looked at DNA polymerase yet. DNA polymerase, its function is very simple. It just makes a copy of DNA. You actually already used DNA polymerase when we went for G-type, right? No, EcoLink, sorry. When we went for EcoLink, you guys use DNA polymerase in your, what's the word? The PCR reaction to generate more functional copies of DNA. That is what its function is. It literally just makes another copy of one strand of DNA. So, how to make DNA? You use DNA polymerase. DNA ligase, that's basically how DNA strands that have been cut are glued back together. Okay? And the endonucleases are the ones that cut the DNA in the first place. So these two go hand in hand together. This one cuts it, this one puts it back together, and this one just makes more copies of DNA. All right? So today's learning intentions, you guys are expected to know the use of enzymes to manipulate DNA, including polymerase. So specifically DNA polymerase and also RNA polymerase, ligase to join DNA, and also endonucleases to cut DNA. Okay, so these are the things included in the study design. So you really need to remember three of these molecules and how they work and what are their features. All right, let's start off with the daily review though. First question, list one factor and describe its effect on enzyme activity. So there are multiple factors that affect enzyme activity. Can you list me one? Let's see. Cyril, think of this as your revision. Temperature. So how would it affect enzyme activity? Describe it. All right. Essentially, I'll give you some hints about this. So enzymes like to be in a very specific temperature range. Okay. So essentially, the enzymes in our body, they specifically like it to be between 37 to about 39, sorry, 38, 37, 38 degrees Celsius. Beyond that, enzymes will experience something. Can you tell me what will happen if the enzyme is exposed to a high temperature, higher than the range? What do you think would happen? Higher than its range. So if it wants to be in this range, what happens when the enzyme is placed at a temperature higher than the range? Yeah, simple as that. Basically, the bonds that make up the enzyme will be broken. So we call that denatured. So make sure you remember that. I'm using this as an opportunity to revise you on the side. Okay, next. So that is if it's too hot. What happens if it's too cold? Sorry, any ideas? All right. Lani, what do you think? Yeah, essentially the enzyme freezes, but essentially because it's frozen, it can still regain its function if it's heated up again. Okay, next question. And non-competitive inhibition affect enzymes. Can I get Ayman? Okay, so tell me about competitive inhibition. Can you describe it in any way? What about it is competitive? So it's in the name itself, competitive inhibition. It is competing with something, right? Why is it competing? All right, let's break it down to the simplest level. Think of an enzyme, right? An enzyme has active sites, substrate binds to the active site, and then undergoes conformational change, undergoes a reaction, releases product. What does competitive inhibition do such that it can't happen? That can't happen anymore. Any idea? Anyone want to help out? Yasmin, what do you think? What is competitive inhibition about? Yeah, basically, it's competing for the active site. That active site, there's two things that would want to bind to that active site, either a substrate or the inhibition molecules. They would want to bind to that as well. Specifically, the competitive inhibition will want to bind to the active sites to block any substrates from binding to that. And what is non-competitive inhibition about that? Can I get Zoe? What do you think?
Non-competitive. It's when the enzyme binds to the allosteric site and
The enzyme,
the
the inhibitor molecule.
site. And so that changes its shape because it's trying to conform to it. So that will subsequently change the shape of the active site so the substrate can't because it doesn't match shape.
Exactly. Okay. So one thing about biology that you guys really need to wrap your heads around is the whole shape conformation thing. Enzymes and a lot of molecules are very shape specific. They can't bind to a location if that shape is not present there. So that's very important to realize in biology, and that's what happens with non-competitive as well. Inhibitor molecule binds to the allosteric site, which is anywhere other than the active site, and then it will change the conformation, cause conformational change of the active site such that nothing else can bind to it. So essentially, it binds to the enzyme. So this is enzyme. The inhibitor molecule will bind to any site that's not the active site, and after it binds, it will cause the active site to change shape such that it won't allow for anything to bind anymore. Okay, that is what non-competitive inhibition is about. Last one. What are coenzymes? Can I get Emelinda? Sorry? Okay, it's another support for enzymes. I need a better description than that. Okay, so let me ask a few questions to scaffold the answer. Does the coenzyme bind to the enzyme before or after the substrate? Before. Okay, essentially, without the coenzyme present, the substrate can't even bind to the enzyme to undergo the reaction. So that's why the coenzymes are important. Can you give me an example of a coenzyme by any chance? No? Okay, anyone else? Abi? ATP, adenosine triphosphate, basically the energy molecule. With the coenzymes, just remember, it can be other molecules as well. But for you guys, what you need to remember is that it is where energy is transferred to the enzyme for it to be able to undergo the reaction. All right. So, which pair of molecules here contain the greatest amount of stored energy? Can I get 10? So think about it, right? Just now we talked about coenzymes and how coenzymes at this level, what you guys need to know is that it transfers energy to the enzyme, specifically in terms of ATP. So we know ATP carries the energy. That's one of the answers. There's another molecule, NADH versus NAD plus. So what is the difference between NADH and NAD plus? What is missing from one and the other? The H, right? Yeah, essentially that H means that it's loaded. That means it has energy loaded onto it. And when it's given away that energy, it will become NAD plus. With ATP, that means it's carrying energy. Once it donates a phosphate group, it becomes ADP, right? ATP and ADP is very simple. There's literally only one difference. The phosphate group, instead of having 3 with ATP, ADP will have only 2. That's why it's triphosphate versus diphosphate. The prefixes, remember, tri, di, mono. So use that as a way to remember. As soon as it donates something, that means it's donated the energy. If it's getting something, it means it's gaining energy. It's loading its energy. So ATP, NADH will be the answer for this one. All right, so getting into today's lesson, so these are the glossary tasks. Please get done with your notes. And also, speaking of which, before we finish class on Thursday, I'll be doing a review of all the summary notes and all of it. And anything that's pending will need to be done over the holidays. And I'll check first class back. If it's not done, then we are in trouble. Okay? I can't keep like letting it slide. All right, so the first one, the endonuclease of DNA as an enzyme, right? So we call endonucleases as being a broad range of enzymes that are able to cut strands of DNA. One such group of molecules is called restriction endonucleases. So essentially, endonuclease is a class of molecules that has a huge range, and one specific group in that is referred to as restriction endonucleases. How those work is how endonucleases work is that they cleave the phosphodiester bonds of the sugar phosphate backbone that hold the DNA nucleotides together. So remember how DNA is built up of nucleotides. That's the monomer. When different nucleotides bind together, that becomes a nucleic acid, which is the polymer. And when they join together, they form a sugar phosphate backbone. Drawing will never be in trouble. I need to redraw this. Is there? So let's see if I can get a picture here. So if you can see here, right, in between each nucleotide, there's a bond that holds them all together, forming a polymer. Okay, that is the phosphodiester bond that is holding them together to be a polymer. What restriction enzymes, what endonucleases do is that they cleave those phosphodiester bonds, and the method in which they cleave is different between different types of endonucleases. So let's look at the one that we're looking at, which is restriction endonucleases. How they work is that they target something known as a recognition site on the DNA to create a cut. So essentially, endonucleases are a huge group of molecules. They work by cleaving the phosphodiester bond. One example of them, one group of molecules under them is restriction endonucleases. How those endonucleases work is by recognizing a specific region known as the recognition site on the DNA to create a cut. So these restriction endonucleases, or we call them restriction enzymes, that's the easiest way you can refer to them. Restriction enzymes, they find these recognition sites on DNA and can either do two types of cuts. One is called a sticky cut, a sticky end. The other one is a blunt end. Okay. As you can see, with a sticky end, there is an overhang of unpaired nucleotides, whereas with a blunt end, there is no overhang. It's simply a direct cut through the DNA strand, whereas with a sticky end, it creates a staggered cut such that there is an overhang of nucleotides. So the difference between them is very subtle, but it creates a functional difference as well, because these nucleotides that have been unpaired, will act as a sort of a glue that is only going to be bound with its complementary nucleotides, right? So essentially, the reason why a sticky end is better than a blunt end is that with a sticky end, the nucleotides will only bind to the complementary set of unpaired nucleotides. So the inserted gene is oriented correctly when manipulating DNA. So essentially, only thing you need to know, with a blunt end, there's no overhang. With a sticky end, there is overhang. And because nucleotides can only bind to complementary, that's why it is the benefit for it. The fact that it will only look for complementary nucleotides means that it will only bind to a region of the DNA that is orientated correctly when being manipulated. So essentially, this reduces the chance of it binding to an unwanted region. because it's looking for a region that has that specific sequence, which more likely means that it's going to be orientated correctly during DNA manipulation. Okay. So these are how they look like, right? So do you notice any pattern in terms of the recognition sites? Look at the recognition sites here: A, G, C, T, then T, C, G, A. A, A, T, 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 A, A. What do you notice here? Yeah, this is what we call as palindromic. So with recognition sites for restriction enzymes, they are more often than not palindromic. So you can always use that as an identification technique, right? So see here, GC, CG, GG, PG, right? They will always be palindromic to each other. That is a very key identification for restriction enzymes. So another detail about restriction enzymes, more often than not, they are extracted from bacteria. These bacteria use restriction enzymes as basically a defense mechanism against viruses. So think of a virus that's invading bacteria. It's going to do massive harm. So what bacteria have done over many thousands, millions of years is it's evolved defense mechanisms against bacteria, against viruses. One of this defense mechanism is using restriction enzymes. So essentially, imagine a virus invades the bacteria. These restriction enzymes will find regions of the viral DNA and just create cuts along it. So what happens if you keep creating cuts along DNA? Eventually, it will become unusable because you don't know what's supposed to be bind to what, and it's just going to create chaos. So that's why it's a very useful defense mechanism against viruses. They are just going to create havoc in the DNA of the virus, making it unusable. All right. So this is what they look like, right? The restriction enzyme binds to the recognition site, recognizes it, will either create a staggered cut like this, where it cuts across, creating overhang, or it will create a cut directly in the middle of the recognition site. So these are the differences. Blunt end, sticky end, cuts in the middle of the recognition site, creates a staggered cut across the recognition site. There will be an overhang here. There won't be any overhang for this one. Yeah.
Why is the overhang better again?
So essentially, the overhang is better because it will look for a sequence that matches its unpaired nucleotides, right? So more often than not, it will find it will find the right sequence to bind to such that it is becoming functional. Whereas with a blunt end, because there is nothing to basically nothing to home it to the right place, anything can be placed in between this, and that might not become a functional DNA copy anymore. Because since anything can bind in between this, it can create a non-functional region of DNA. But with this, since it is sticky end, it will look for a complementary strand, and more often than not, that allows it to make sure that the gene is orientated correctly when the DNA is being manipulated. So essentially, it's a way that it can remain functional when DNA manipulation occurs. So DNA manipulation, we use restriction enzymes to cut regions of the DNA to possibly insert another gene or for any other purposes. And when we're done manipulating it, we want to make sure that they join together back more or less well. So this sticky ends will allow them to join together such that it's orientated correctly, making sure that the DNA still remains functional even after being manipulated.
Don't want them to find a complementary. You don't want them to fix themselves in the
So essentially, this cut will happen, but it won't fix itself without the next enzyme, ligase. So with the bacteria doing the cuts, even if you do this cut, it can't join back together if you don't have DNA ligase, right? So the bacteria essentially won't make DNA ligase in that situation. It'll just create cuts everywhere along the virus, and then that'll essentially make it non-functional. All right. So now for the second enzyme, ligase. This is the gluer. Is that even a word? The gluer. Never mind. The glue. Okay. So if endonucleases were the scissors that cut the DNA, ligase is the glue that joins two fragments of DNA or RNA together. So it's essentially a molecular glue that joins strands together. Okay. This happens through a simple enzyme reaction where it catalyzes the formation of phosphodiester bonds between the two fragments. So remember that this is an enzyme. Enzymes reduce the activation energy. Essentially, they catalyze a reaction, right? So that's what's happening here. The enzyme of ligase will catalyze the formation of phosphodiester bonds between the two fragments to merge them together. So if I ask you, is there any situation where the phosphodiester bonds can form by themselves? Is that possible? Remember, with enzymes, all they do is that they reduce activation energy, right? So essentially, even without an enzyme, there is a possibility the reaction can occur. It might take very long, it might take a lot more energy, but there is a chance it can occur. That's the same thing here. Since ligase is an enzyme and all it does is that it catalyzes the formation, there is still a chance it can happen by itself, even though it's a very low chance, because essentially the DNA needs to be held together in a very correct formation, and then only that reaction can occur. So the only thing holding it in place like that is the ligase. So essentially, it's very, very low chance it can happen, but it's not zero, because enzymes only catalyze reactions. They do not create reactions on their own. So we need to make that distinction. The difference between catalyzing a reaction and creating a reaction. Enzymes are all about catalyzing it, meaning they lower the activation energy required. So there are two types of DNA ligase that you guys need to know about. Two types of ligases that you need to know about. One is DNA ligase, and the other one is RNA ligase. And it's pretty obvious which ones they work on. DNA ligase works on DNA, RNA ligase works on RNA. And since this is basically building a bigger molecule, taking two smaller molecules and building it up into a bigger molecule, it is referred to as an anabolic process, right? Anabolic is about building up, catabolic is about breaking down. So this is an anabolic process, and it works by letting the substrate, which are the sugar and phosphate groups, to bind to the active site, and then eventually facilitating the reaction to occur where they join together to make one complete strand. Right? Any questions about this? Right? First thing to remember, DNA ligase is everything we're looking at today is an enzyme. So essentially they catalyze reactions. So they can happen theoretically, but it's a very, very low likelihood, because the conditions required for the reaction to occur are such that it's not going to happen by chance. It requires enzymes to happen, but regardless, since it's an enzyme, you need to make that distinction. Okay? So with the ligase, it catalyzes the formation of phosphodiester bonds. So that's why ligase and endonucleases come hand in hand, right? Endonucleases are about breaking that phosphodiester bond, whereas with ligase, it's catalyzing the formation of the phosphodiester bonds. Okay, that's why they will always go hand in hand. You can always remember one and the other based on their opposites, right? Ligase is about building up, whereas endonuclease is about breaking down. Any questions? No?
Can you expand on the substrates thing?
Okay. Essentially, remember how enzymes work? Right? Yeah. So they have active sites, right? So enzymes have active sites. In terms of how DNA ligase works, is that the ligase binds together with the DNA, and then the sugar group of a nucleotide of a nucleic acid and a phosphate group of the nucleic acid will bind, joining together at this region, and then eventually the reaction to join together the bonds will happen, creating one strand. So the active sites will allow the binding of sugar and the phosphate group, and then only that reaction can occur. Sorry, and then only it can catalyze the reaction, allowing for one complete strand to occur. Let me try to zoom in. Yeah, essentially, at this point where the cuts have happened, this is where the sugar and phosphate groups will bind to the active sites, and then allowing for formation of phosphodiester bonds to occur. Any questions? All right, moving on. Last one is polymerases, the creator. Seems like overtly extra, but couldn't think of anything else. The creator, because it basically creates functional strands of DNA or even RNA. Okay, so there's two types of polymerase that you guys have looked at so far. One is RNA polymerase. That's the one you're very familiar with. It makes mRNA, right? It's used for protein synthesis. That's the one we've even practiced using as well. It binds to the promoter region and then moves along the DNA to make a functional mRNA copy. That is what we know about RNA polymerase. DNA polymerase, on the other hand, is about making a completely new DNA strand out of a copy of a strand that they already have. So there's DNA copy, DNA polymerase binds, creates another strand of DNA from that original copy. Okay, so certain things are required for DNA polymerase to function. One of them is primer. Okay, you guys, I'm not sure how much you remember from your EcoLink excursion. Everyone went, right? Everyone in this class. Who didn't go for the Aiman? Okay. So now is when you realize how much you've missed out. All of them got to use DNA polymerase during the excursion to create functional copies of DNA. Essentially, how it works is that a primer, let me zoom in the photo. Right? So essentially, we have a DNA strand. Okay, the bonds between the nucleotides have been unwound, and there will be a space for the primer to bind. Okay, once the primer has bound, it will bind specifically to complementary sequences of the template strand. Remember, the template strand is the one that is used to make a complementary copy. So that's why we call it a template strand. The primer will bind to the template strand, and once it's bound, the polymerase will bind next to the primer itself, allowing and then facilitates the formation of another copy, a new strand that is complementary to the template strand. Okay. So with this, it binds to the primer and then it reads and synthesizes a complementary strand of the template strand in a 5 to 3' direction. Any questions about this? Oh wow, we're doing too good for that. Right? So essentially, the three enzymes that you guys need to remember, restriction endonucleases, or you guys can just call them restriction enzymes. I know the word endonuclease is a bit annoying, so just call it restriction enzymes, ligase, and polymerase. Okay? With a restriction enzyme, it's about cutting DNA or RNA at recognition sites. With ligase, it's about joining them together to form one complete strand. With polymerase, it's about amplifying or multiplying sections of DNA or RNA. Okay, one is about cutting, the other one is about gluing, this one is about copying and pasting. Okay. All right, so this is a question about restriction enzymes, and this is actually a very common type of question that they're going to ask. Remember that restriction enzymes work by finding a recognition site within the DNA sequence, and then only it does the cut, right? So, actually, let's talk about the names of restriction enzymes. I don't think I mentioned that. All right, essentially, with the names of restriction enzymes, it will always relate to where they're coming from. Okay, remember that restriction enzymes, we are extracting them more commonly from bacteria. One of that So one of the example is EcoRI, and that is extracted from E. coli. So that's why it's called EcoRI restriction. So anyway, it's EcoRI from E. coli. So with this one over here, you can see there's a bunch of names here, right? EcoRI, HINC2, PST1, HINC2. Each of these are extracted from a different bacteria, and such they are named differently. Okay, so one plasmid was mixed with the restriction enzyme EcoRI, BAN-HI, and HINC2. HINC2. Which of the following shows the number of restriction sites that have been cut and the resulting number of DNA produced? Okay, so essentially, these things that you see here, these are the recognition sites. Okay, if these recognition sites allow for the restriction enzyme to function, how many strands are they going to make? Okay, first question, how many restriction sites are cut, and then how many DNA fragments will be produced? Okay, let me zoom into the picture so you guys get a clearer sense. All right, so there's 3 restriction enzymes that have been mixed, EcoRI, and HINC2. There are multiple recognition sites. So think about where are the cuts going to be made, and then how many strands that's going to make in the end.
Because it has to kind of polymerase.
Essentially, here you wouldn't concern yourself with that because here you don't even know what the DNA sequence is. Here they've already given you it as a recognition site, so you don't need to concern yourself about nucleotides or whether it's palindromic. It's already ready to be cut, essentially. All right, can I get who would like to volunteer to answer this question? Any brave volunteers? Khadija, what do you think? All right, let's go through this question together, right? You got 3 restriction enzymes. Do all 3 have recognition sites on the thing, on the plasmid? Yes, right? How many recognition sites are there in total that are going to be recognized by these 3 enzymes? EcoRI, BAN-HI, and HINC2. 1, 2, 3, 4. So essentially, there are 4 recognition sites that the enzymes can bind at. So EcoRI, BAN-HI, HIC2, HIC2, HINC2. Yeah, the names are the most annoying part. So there are 4 different recognition sites. That means there's going to be a total of 4 cuts that are made, right? If there are 4 cuts that are made, how many strands will then be produced? Okay, think about that. 4 cuts, how many DNA fragments are produced? It's not too complicated actually. Just think of it as a diagram here. You cut one here, cut one here. How many fragments have I created now? No. Think 2. Essentially, if I separated these 2, I would have one that is this long and then one that is this long, right? If I do this. So think about that in terms of that plasmid, right? I'm making a total of 4 cuts. So how many strands are going to come out of that? So a lot of people get confused with these kind of questions because they essentially think making one cut will create a fragment. But when you make a cut in a plasmid, you're not even making a fragment. You're just cutting one region such that it becomes one long strand. A plasmid is circular. So when you do that cut, all you're doing is making it into a strand. You're not creating any fragments. You need to at least create 2 cuts to create one fragment. So 2 fragments out of that. So if you have 4 cuts, what do you think will happen? Okay, let's model it, shall we? So create a circle, and then let's model all the cuts. One will happen here, one will happen here, one will happen here, and another will happen here. So how many strands are created? So is this a strand? That's one. Is this a strand? That's two. Is this a strand? That's three. And this is a strand. That's four. So you can see here that 4 strands will be created. Okay. Essentially, a technique to do this is that you have a question paper, just write where the strands will be, right? So essentially do what I did. Just number the strands every time there's a cut that happens, and you will be able to find out very quickly. If you try to work it out in your head, it will take more time. Trust me, something about doing this in your head always takes longer. Just model it using a drawing, and it's very quick to get done. Okay, so make sure you remember that one. All right.